everyone. Welcome to this new episode of our Student Spotlight series where we're able to showcase some of the work our past students have done. I'm Charlotte, one of the outreach coordinators here at CCIR, and it's our pleasure today to have Diya with us, who did our one-on-one mentorship. So firstly, can you just introduce yourself? So tell us where you're from, what year you are in school, and then what made you seek out CCIR?
Hi, so my name is Diya. I've just completed my 11th grade. I live in Bangalore, India. And the reason I chose to seek out CCIR is because I want to pursue research and I really wanted to have the opportunity to write my first research paper in high school. So I took a look at a couple of the programs and CCIR's outline and its brief really suited my preferences, which is why I chose to do this.
Great. And then can you give us a general overview of what your course was about in the one-on-one mentorship and what you looked into?
So my interest is immunology, and I've been interested in this for quite some time since the 10th grade. So when coming into this program, I wanted to look further than what I already knew. So I was already quite clear with the fundamentals, with the innate immune response, the adaptive immune response, a little bit about vaccines. But something I didn't know was immune regulation. And I chose to focus on a subset of T cells called regulatory T cells, which play a really critical role in the regulation of the immune response. And my paper is about how these T cells actually demonstrate different functional properties in different tissues of the body. Apart from that, I also did some data work where I investigated the role of these T cells in chronic viral infections as well.
Interesting. And then had you had formal experience in school with immunology before, or was this kind of your first time really getting into it in a formal setting?
So in school, we had to do a project, and I chose to do immunology. So that was sort of my introductory experience to immunology, but it wasn't a formal teaching setting, and I didn't have a mentor who was there to guide me during that time. So yes, this was my first formal research experience with immunology.
Nice. And then now we'll shift to discussing your specific research paper. So can you give us a general overview and then how you decided on this topic?
So when I first connected with my mentor, he suggested that I pick one existing research paper and I summarize it, and then we can find what interests me. And I did exactly that. I found a research paper about the role of these different T cell subsets in mucosal tissues. And there was literally one tiny paragraph about regulatory T cells in that entire paper. But when I was presenting it to my mentor, and this was a 20-minute long presentation, um, he could sense my enthusiasm for that one slide where I covered that topic. And when he asked me, what was your favorite slide out of everything you've just presented to me, that was the first thing I said. And he agreed with me. So that was really how I discovered regulatory T cells. My paper is about how this group of T cells, which is supposed to have one function, which is to mediate the immune response, actually seems to showcase different functions, sometimes contradictory functions in different tissues of the body. And throughout my paper, I look at different factors, different location specific factors that might explain this strange yet fascinating phenomenon. And then I also look at some real life patient data and I analyze how these Tregs behave in chronic viral infections, which take place over days and months and sometimes even years.
Interesting. And what was your process of finding the data? Kind of how niche is this topic, and what was your process of analyzing the data?
So I would say that compared to I mean, immunology itself is quite niche, but compared to other immune cells, regulatory T cells is something that's quite recent. So yeah, it was a bit of a struggle to find the right research papers. Another thing is that a lot of research papers are quite specific. So they'll focus on only one or two tissues, or maybe only one or two factors. But I wanted to do a, a macro view, right? A general overview of everything that there was. I didn't want to focus on one or two tissues. I wanted to pick out examples from different tissues so I could create a sort of cohesive argument. So I started with a preliminary lit review, and then I structured my paper in the most logical way that really gave a comprehensive overview of everything that was out there so far. And then I looked through paper databases and I annotated the papers, and I built up my lit review. For the data section, my mentor was really gave me some invaluable guidance for that. So this was my first time doing any sort of data. I actually had to take genome sequences of viruses from an existing paper, and then I had to use some computational tools to sequence them and then find some sections of those viruses that are prone to mutation. And then I checked if those sections fell into something called T cell epitopes. So an epitope is a section of something foreign, like a virus, which interacts with immune cells. So if these variable sections in the virus fall in the same place as these epitopes, it could suggest that the immune cells, like regulatory T cells, are driving the mutation of these viruses. So I did this for 8 different patients who had who were infected at different points of time just to get some variety in my data and for different durations. And what I found was that there were actually these mutations in these T cell epitopes. And the interesting thing was these mutations were accumulating over time. So in day 0, if you had 2 mutations, then maybe in day 600, you would have 15 mutations or 20 mutations. So this led me to believe that these regulatory T cells are actually driving the accumulation of these mutations. And then I had to investigate why this might happen, right? And I found some really interesting literature that said that on one hand, the mutations may help the virus evade the immune system, right? So this could be harmful because it could allow viral persistence in these patients. It could allow you to be sick for a longer period of time. But on the other hand, this could actually help to suppress an immune response that would otherwise be harmful for the patient. Because sometimes what you really have to fear is your own immune system. It can be so strong, right? So there's this intricate balance that has to be maintained between protecting yourself from viruses or bacteria, but also protecting yourself from your own immune response, which, if it goes overboard, can be very deleterious to you. Yeah.
That's super interesting. Um, yeah, wow. And so in terms of kind of your findings, so you found this discovery. Is this something that you were expecting to find? Was this completely out of the blue? And what are your kind of thoughts for the research moving forward?
So I did have a hypothesis. So I did expect to see at least some mutations that fell in these epitopes, but I didn't expect to see sort of such a consistent pattern in every single patient and in such a variety of patients. So I actually graphed it, and you can clearly see like an upward trend as you go over time. I think that in terms of my future plans, there's not a lot of research about this, so I can't make a definitive conclusion. And then there's also other things to consider, such as the virus, it has its own background mutation rate, right? It mutates on its own. So what if these mutations that I'm discovering are actually just happening on their own and they happen to fall in these T cell epitopes? So I can't say anything with certainty, but it does look promising. I think that with viruses, especially, there's not a lot of research that's focusing on T cells, much less regulatory T cells, right? There's a lot of focus on antibodies that's out there. And if we can decode these different functions in different tissues of these regulatory T cells, that could open up a whole new option for therapies, right? That are targeted for infections or diseases that are in specific tissues. And this is really exciting. And yeah, I'm really excited. I'm going to keep up with the literature. I'm really excited to see what's going to happen. Yeah.
That's so interesting, and it must it's so great that you're able to kind of contribute to this field that is this field at such a young age. And then just in terms of kind of your publishing, so I know you're at the beginning stages of the publication process, but can you just describe the support that you've received from CCIR up until now and kind of what your steps for publication are going forward?
Yeah. So I think CCIR has been very helpful at every step of the process. While I was doing my research, it was really useful to have a literature. CCIR was able to provide access to some papers which I wouldn't be able to access otherwise. And they were checking in on every step of the process, which was really good. I sent in my paper a few weeks ago and they sort of went through the entire thing and they provided some feedback for me so I could touch it up a little bit. And now they have provided a comprehensive list of conferences, symposiums, and journals that I can apply to, which have been it's a very thoughtfully curated list specific to my own research topic and what I am looking to showcase. So I'm very excited for the journey ahead. They're happy to help me with personalizing my paper with the format or the references that are needed for each particular journal, and they provided all the deadlines for me. So I think that will be the next step to um, get my paper published or to hopefully present it somewhere.
Nice. So now we're just going to shift to discussing your general experience with CCIR. So can you tell us what your experience was like in the program doing a high-level research project like this as a high school student?
So I think it was incredibly exciting. I I was really happy with the fact that I got to meet my mentor before actually closing the deal. And I was matched with a mentor who really understood my passions, and we were aligned on the our how we thought and our interests. And he was really able to give me a very um, realistic overview of what it means to work in the field. It was really nice to have a personalized one-on-one experience with my mentor. And it was also very flexible. So I live in India. There are issues with coordinating across time zones, but CCIR was really, really helpful with that. And I also had a lot of other commitments with my exams, with travel, but they were really flexible. I was able to stretch out the program for however long I wanted. I was able to take my time to work on my paper, to do the edits, to get some feedback, everything. So that was really, really great. And I think it's so rare for a high school student to actually develop a rapport and build a relationship with a mentor like this. And I'm very, very grateful that I got the opportunity to do so.
That's great. And I think you brought up a good point that it really is a relationship that you build as
Yeah.
opposed to kind of this the traditional teacher role where they're just passing information on to you. And in that case, can you describe us a little bit more about that, specifically with how your mentor helped guide you through? I know you mentioned earlier that they helped you really recognize what you wanted to research. How did they help guide you through that initial learning stage and into the research process?
So yeah, of course. So my mentor was Dr. Ruiz, and he was from the University of Cambridge. So he was really helpful with, I think what I appreciate the most is, like you said, it wasn't really a teacher-student experience. It was more like we were having an organic conversation. And he was just asking these thought-provoking questions, and then we were really having a discussion, right? Rather than it being a one-sided transfer of knowledge. He was very inspiring for me because his career path is sort of something that I would love to follow. And he really taught me about some tools and some skills that I need to develop if I want to go into this field. So, especially with the computational work, which I'm not exposed to at all in my school environment, I now understand what else I need to do independently to get to that stage where I am prepared to go into the professional world of research. I also think that he was very flexible with his feedback. He was willing to give as many rounds of feedback as possible. He never got sick of reading the same thing again and again. He was really, really kind in that sense. And I'm very grateful for his guidance.
That's great. And then I know we spoke before about how immunology is still kind of a growing field. What was it like being able to take such a niche kind of specific course that you wouldn't necessarily have access to in high school and even sometimes in your first or second year of university? What was that like?
I think that I was speaking to my mentor about this in the beginning of our program as well, and he told me, you've chosen one of the most difficult things to work with, but also one of the most exciting things to work with. And I was like, yes, I absolutely agree, but this is just something I'm so passionate about that I would love to go forth with it. And even my mentor was really passionate to go to research something like this because even he hadn't been exposed to this particular topic. But it was really nice to get a fresh perspective from him about the virology aspect of it, which was very invaluable for my research. And yeah, I think that it's incredibly rare to find someone, to find a mentor who's willing to guide you on something that's so niche, so specific, and yet be such an incredible presence, an incredible guide in this research process. And I think it was incredible to have a platform somewhere where I can feel safe and I can feel like I'm able to experiment. I'm able to take my time and I'm able to do exactly what I want. I don't have to compromise because it's so one-on-one and it's so personalized. Yeah.
So great. Yeah, I think that's definitely one benefit about the one-on-one mentorship is that you really are the driver of what you learn, what you research, where the course goes. So it gives you that freedom to make sure that you are really going down the path that you want. Just a few more questions for you. The first one, do you think this course has given you any insights into your future? Has it informed or maybe shaped how you think of your path moving forward?
Yeah, definitely. I think that it's really solidified what I already wanted to do, and it's reaffirmed my passion for biology, chemistry, immunology. So I had some discussions with my mentor as well about my future, and I'm planning to pursue an undergraduate degree in biochemistry or molecular biology, and then after that, hopefully, a master's in immunology. And this experience has definitely opened the doors for me into the research world and what it is to be a researcher working with real world patient data.
Nice. And then my last question for you, what would you say to someone who asked you what the value of doing a project or a program like this is, especially for a high school student?
I think that definitely if you have the time for it, if you have the resources for it, and if you have a passion for research, you should take a go at it. You should apply. The entire process is incredibly smooth. It's incredibly exciting. You're going to get to meet someone or some people who you haven't met before. You're going to get exposure to the real world and research experience. It's not as easy as it sounds to do something on your own, and it's incredibly useful to have a mentor. And like I mentioned before, it's not only a mentor for this one paper that you're doing, it's a lifelong connection that you're going to be able to build. You're going to be able to have a rapport with someone, a relationship with someone that's going to last for a long time if you take the effort to make it last. So I think that to anyone out there who's looking for something like this, you should definitely go for it and take a chance. Yeah.
I think that's great advice. So that brings us to the end of our spotlight interview. Thank you so much for chatting with me, and congratulations again on your nomination to our series.
Thank you.